So good afternoon, everyone. Welcome to our last student research presentation on diet composition and diet quality. My name is Yi Zhao. I am a PhD candidate here at Tufts Freeman School of Nutrition, and I will be your moderator for this session. Today, we have such an international group of students presenting today, so let's begin. First, let's welcome Ms. Wiksa from Management and Science University, Malaysia.
A very good day to everyone. My research is on the interaction effects of food security and diet quality on weight changes among working women of reproductive age during the endemic phase of COVID. I have no conflicts to disclose. So the pandemic-related economic downturn has affected food security, reducing the access to adequate and nutritious food. And we understand that compromised diet may lead to nutritional consequences, such as weight gain and obesity or overweight over time. The study was the first of its kind to assess the interaction effects of food security status and diet quality on weight changes among working women of reproductive age. So this was a cross-sectional study involving 263 Malaysian working women. The recruitment of respondents were conducted in person by using a combination of purposive and convenient sampling. Socio-demographic information and pre-pandemic weight were acquired through self-reports. Body weight and body height as of the time of data collection were measured by using weighing scale and steadometer respectively. As for diet quality and also assessment of food security status, diet quality questionnaire and food insecurity experience scale, FIES, were adopted for those purposes. These are the key findings. So food insecurity was a concern to 35.7% of the working women who mostly belonged to the mild food insecurity category. As for diet quality, 82.51% managed to achieve the minimum dietary diversity for women, or in short, MDDW, which signifies the consumption of at least 5 healthy food groups out of 10. As for absolute weight gain, a total of 64.3% reported an average weight gain of 4.36 kilograms in just less than 3 years into the pandemic. There was also an increase in the prevalence of overweight and obesity during those periods. And the results from two-way ANOVA, as represented by the table on the right, shows the mean difference in weight change by food security status and the attainment of MDDW during the endemic phase. So it was reported that only poor diet quality was the significant contributor of weight gain and not the synergistic effect of both food security status and diet quality, whereby those who did not achieve the MDDW has put on an average weight gain of 4.02 kilograms, which was significantly higher when compared to those who did achieve the MDDW. And in terms of the interaction effects, though it was not significant, it was reported that those who were moderate to severe food insecure and failed to achieve the MDDW at the same time were reported to have the highest average weight gain of 4.85 kilograms during the endemic phase of COVID. So in short, employers should ensure the implementation of workplace-based initiatives to nudge working women towards healthier eating. And the initiatives are a healthy cafeteria, vending machines, or on-site cooking demonstration, or even yearly nutrition consultation, just to name a few. And in doing so, we hope to help these people to better regulate their weight. So this research reflects the compliance of Sustainable Development Goal 2, ending hunger, as well as Goal 3, good health and well-being. And last but not least, I'd like to acknowledge the contribution of all respondents for their voluntary participation in the study. Thank you.
Thank you, Ms. Wiksa. And the third presenter is Belen Beltramo from Maastricht University, Netherlands. Welcome.
Thank you. Thank you very much for the opportunity to be presenting today. My topic is about a comparison between European food composition tables and how the differences between the tables could affect the use of nutritional health claims. First of all, I don't have any conflicts of interest to disclosure. And to start on the topic, in the European region, fresh produce, and here we talk about mainly fruits and vegetables, that are sold by the kilo or by bulk, they are not required to declare the nutrient information, so the back of pack information, but they can still make claims. So these claims, the main source to make these claims are national food composition databases, but at the same time, there's a lack of harmonization between countries. And also the trading of these items it's very open in the European region. So you could have, for instance, an apple that from Germany that it ends up in Italy, and that apple can use the food information from Italy to make a claim. But are these this data composition, is this data composition comparable? Is it the same? We still don't know. So as a solution, we came up with this study where we compare the food composition table of main trading producer countries of fruits and vegetables at the absolute level, but also the practical level, so that the use of nutrition and health claims. And at the end, all with the aim to give recommendations to producers and also to know the state of art of the current data that we have here in the region. To do this, we sample nutrient content from fruits and vegetables from national databases from the 9 countries. These are the main producers of fruits and vegetables in the European region. We use the platform Eurofear, which shares food composition tables from most countries in Europe. We selected the most traded fruits and vegetables, and we analyzed the micro and micronutrients that are both most affected by external conditions, so the cultivar, the type of cultivar, the climate, and other conditions that can affect that, but also that are mainly used or subject to food claims. We use 2 cutoff points at the 50% and the 30% of the reference values, which are the cutoff points that are used for source of high-in claims. And we compare the data reported by each country. As key findings, I will be presenting you the data from vegetables and at the source of claims, because those were for those items were the ones that we find more differences. So we saw that there was low discrimination at the cultivar level. So there's not many countries that also report information at the cultivar level. So it's more like a generic report of information, where there at the cultivar level, there's a lot of information that might be not communicated. We also didn't see too much data on the sampling season, and we do know that there are differences between the season, mainly micronutrients. And we also saw that vegetables were the ones that differ the most compared to fruits. So that's what I'm presenting here. And we saw that the vitamins presented higher differences compared to minerals, and especially vitamin C. And vitamin C and folate were the ones that were more different. And for instance, to represent it with an example, we saw that spinach that was reported in the data food composition tables from Germany, the Netherlands, and Belgium, which are 3 neighboring countries. 2 countries could make a claim, so Germany and Belgium, but not the Netherlands. But we still don't know if that difference is really there, or if it's some differences at the way each country report the results and how they measure the results. So all these results that we obtain, what we would recommend producers is to use databases as a sanity check, as a yes, no, to continue analyzing the food composition in their real samples, to advance into making a claim or not. And but more also, we think that more information on biodiversity is needed, but now we still encourage producers to measure themselves, because we know that there are no programs looking into this at the moment in Europe. So yes, these are the main results that I have to present to you. And acknowledgements, I want to recognize the European Regional Development Fund for supporting my PhD, and my supervisors and colleagues at UM. Thank you.
Our next presenter is Tasija from University of Rhode Island. Welcome.
Hi, everyone. My name is Qisi. I'm a PhD student at the University of Rhode Island. It's my pleasure to be here with you today. I will be talking about a study that I did where we looked into replacing ultra-processed food with minimally processed food on weight change in a secondary analysis of the Pounds Law study. It is well established that ultra-processed food intake is associated with excess weight gain and development of obesity. However, it is unclear if replacing ultra-processed food with minimally processed food would favorably influence weight loss in an energy-restricted environment. To address this gap, we looked into these associations in the Pounds Law study. Briefly, the Pounds Law study explored the effects of 4 energy-restricted diets of varying macronutrients composition on weight loss among individuals with BMI over 25. For our study, we looked into a subset of participants who have at least 2 days of food diary data at baseline and at least 1 day of 24-hour dietary recall at 6 months. At baseline, the participants were on average 53 years of age with a BMI of 33. So participants' dietary data were coded using FNDDS before being classified into one of those 4 NOVA groups to estimate percent energy intake in each group. For those not familiar with NOVA, this is a framework of grouping foods based on the extent and purpose of industrial processing. And for analysis, we used substitutional models to see if replacing ultra-processed food with minimally processed food would differentially associated with changes in both body weight and waist circumferences between baseline and 6 months, adjusting all the covariates listed here. What we found is that isocaloric substitution of ultra-processed food with NOVA group 1 and 2 foods was associated with greater weight loss in the Pounds Law study, a energy restricted environment. More specifically, replacing 10% of ultra-processed food with NOVA group and 2 foods was associated with a half a kilo greater weight loss with a p-value of 0.02 and almost 0.5 centimeter greater reduction of waist circumferences with the p-value of 0.07. So this studies gave us additional reason to explore pathways linking ultra-processed food and regulation of body weight beyond energy intake. I would like to acknowledge all my co-authors. Thank you for listening. I'll be very happy to answer your question during Q&A.
Thank you, Qisi. And our next presenter is Odessa from University of Washington, Seattle.
Hi, thank you for having me. Hi, so my name is Odessa. I'm a recent graduate from the School of Public Health at the University of Washington. And today I'll be presenting research coordinated and directed by Anna Valencia and David Marcinek, which I was an assistant on the role of mitochondrial function in diet-induced weight loss. I'll go to a so some of the background to this research, as you're all aware, weight loss interventions are common in both public health and healthcare spaces. And despite their prevalence, weight loss attempts have shown to result in a weight loss plateau and weight change that precedes eventual weight regain. So the goal of this research was to better understand how mitochondrial dysfunction at the plateau and how responses to energy demand may be altered at this state. So the aims of this research were to, 1, establish a model of weight loss plateau using adult mice with diet-induced obesity, and 2, to determine if mitochondrial metabolism in peripheral tissues was altered at the plateau. So the way we want to look into these questions is we induced obesity in adult male mice using a high-fat diet for 12 weeks. Following this period, the mice in 3 weight match groups underwent one of 3 different weight loss strategies, one of which was a 20% caloric restriction on a high-fat diet. Another was a 20% caloric restriction on a low-fat diet. And the third group was an unrestricted low-fat diet. Following these weight loss periods, we assess mitochondrial function using high-resolution respirometry. And here we looked at the oxygen consumption in permeabilized liver cells to determine maximal capacities of oxidative phosphorylation and ETC to determine how responsiveness, or electron transport chain, to determine how responsiveness to energy demand may be altered at that plateau state. So the first of our key findings was the variability in the propensity towards weight loss based on the diet type. So you can see in figure A the different weight loss trajectories by the different diets. And in figure B, we described the plateau using the slope as a change of body weight more than negative 0.05 grams per day. As you can see in the high fat group labeled in the pink color, they reached a plateau in 10 days, whereas the low fat diet group in blue did not reach a plateau in this 2-week study period. However, they did lose more weight than the high fat group, which was attributed to a greater loss in adiposity. Further, the separate cohort of mice on the low-fat ad libitum diet, shown in the teal color on the bottom, they reached a weight loss plateau after 35 days, and at this point, their weight matched the healthy weight control group. Secondly, we use correlations to determine which aspects of mitochondrial function were associated with both the magnitude and maintenance of weight loss, shown in figure C. So here you'll see the magnified boxes, the maximal capacity of oxidative phosphorylation were higher in mice that lost less weight. And similarly, the maximal capacity of ETC was higher in mice that were more weight stable. Now, most notably, we found that the most robust change in mitochondrial function with weight loss was a reduction in ADP sensitivity in liver tissue. And this was indicated by a higher EC50, shown in figures both D and E, across all weight loss groups, regardless of the weight loss type, compared to our healthy weight control groups. This suggests that mitochondria in the liver became less capable to respond to energy demand, despite increased respiratory capacity, as I previously discussed. Now, ultimately, these findings highlight the potential benefit of a low-fat diet in the witness delayed development of a weight plateau, potentially reducing the likelihood of weight regain in the future. Yet this requires further study. And finally, our results suggest that there are mitochondrial deficiencies during this weight-reduced state that are not accounted for by respiratory capacity. I'd just like to thank you all for your time today, and Anna and the lab for allowing me to contribute to this experience. Thank you.
Great, thank you very much. Our next presenter is Yongyi Pan from Tufts University.
Hi everyone, I'm Yongyi from Tufts University, Freeman School of Nutrition Science and Policy, and happy to introduce our research related to consumption and Alzheimer's disease risk in Rush cohort. And to begin with, we have no conflicts of interest with other teams. And our aim is to examine the relationship between consumption and the risk of Alzheimer's dementia incidence in a cohort of community-only older adults. So brief introduction to Rush aging cohort is a cohort beginning in 2007 and end at 2013. And in our screen of participants, we have 1,025 participants who are 83 at the baseline and have at least one completed frequency questionnaire. So we have a population of participants with average age of 81 years old and an average of 7.2 years average follow-up. And we get consumption, a categorical variable from the food frequency questionnaire. And we also got total calorie intake, strawberry intake, and other food intake from the questionnaire. We also get other demographic information, health status, and health behavior from the same cohort. And then we use Cox proportional hazard model to assess the incidence of AD with consumption frequency. And all the adjusted covariates are in the blue and the green frames. Our key findings shows that on the left chart is the survival curve, and we found that higher consumption are population with the higher consumption are having a higher risk of survival. And on the right part, we found that higher consumption are associated with the lower risk of AD diagnosis. and the results are stable in both unadjusted and adjusted model. So we conclude that more frequent consumption may reduce the risk of Alzheimer's dementia in the elderly population. And we want to thank you for all the participants in the Rush cohort and also the collaboration with the teams. And I'm happy to receive any questions, and thank you for listening.
Great, thank you. Our next presenter is Tasija Karosas from Tufts University.
All right, hi everyone. My name's Tasija Karosas. I am an MS and PH candidate at Tufts University, and today I'm going to be just discussing the association between dietary choline intake and Alzheimer's dementia risk using the Rush memory and aging cohort. So it was the same study sample as what Yongyi just presented. Start off with, there were no conflicts of interest in this study. To dive right into the objectives. So based on the data that Yongyi presented earlier, we wanted to isolate the choline from eggs and other dietary components to assess the risk of Alzheimer's disease in a cohort of community dwelling older adults. So the methods we used pretty similar to study population selection. We use the Rush aging, to what Yongyi said, we use the Rush aging and memory cohort. They had to be free from Alzheimer's disease at baseline, and they had to have completed at least one food frequency questionnaire. The mean age was 81.4 years, and the follow-up time was 7.2 years. Choline was then quantified and split into 5 different quintiles, or 5 different categories. So there were quintiles. The first quintile was negative or below 200 milligrams per day, and then it increased by 50. And then the last quintile was greater than 350 milligrams per day. And the outcome that we assessed was clinical diagnosis of Alzheimer's disease. And we use Cox proportional hazard models to assess Alzheimer's disease incidence on choline intake, and adjusted for a variety of different variables, which you can see in the right-hand part of the screen. So the key findings, just what the results told us, is that compared to the lowest choline intake group, which is represented by the blue line, as you can see in the figure, quintile 3, quintile 4, and quintile 5 all significantly reduce the risk of Alzheimer's disease. And the largest reduction was seen in the highest choline intake group, or highest choline intake group, that saw a 55% reduced risk of Alzheimer's disease compared to the lowest choline intake group. So we can ultimately conclude that higher choline intake is associated with reduced risk of Alzheimer's disease. Just a quick acknowledgement, we were able to get the choline qualifications from the Egg Nutrition Center and the Think Healthy group. And then of course, I want to thank Dr. Taylor Wallace and Mei Chung for just being very involved in helping me through this study. So thank you.
Thank you. Our next presentation will be pre-recorded by Yutong Chen at Tufts University.
Hello, everyone. My name is Yutong. I'm a first-year PhD student at Tufts University, Freeman School of Nutrition Science and Policy. I'm sorry I cannot do the live presentation today due to other commitments for TNDS, but please email me if you have any questions. I want to talk about my study, Comparison of Dietary Intakes in the Global Burden of Disease and Global Dietary Database, a case of sodium consumption. There is no conflicts to disclose. In this study, we aim to explore the degree of agreement between the two most comprehensive databases, Global Dietary Database and Global Burden of Disease in estimations of sodium consumption. We also aim to explore the gaps between current consumption and the recommended intakes, 2.3 grams per day. To prepare for the dataset, we compare the definitions of sodium in both datasets. We also align the dataset based on population age, units, time, and ban country. Our final dataset contains sodium consumption data for 4 years and across 169 countries. For data analysis part, except for the descriptive statistic, we also use the Belen-Altman plots. On the right side, it is what the BA plot look like. The x-axis is the average and the y-axis is the differences. If GDD and GBD are completely same, then all the points will be on the y equals to 0 line. Here are our results. Panel A and C are scatter plots, and panel B and D are Belen-Altman plots. Left panels are original data, and right panels are log transformed data. We also point out the countries with extreme values in each panel. The green zone is the reference level, or can be called a safe zone, which means the countries in that zone has consumptions below 2.3 grams per day. We also found in GDD, there's 123 countries, and in GBD, there is 104 countries overconsumed sodium. We also compared the global mean using the formula in the slide and detected 36% in difference. The most interesting finding is we found 147 countries that at least in one time point were at or below the reference level in GDD, but none of them were as such in the GBD. Therefore, there is a large discrepancies between these two databases, and we think more accurate data should be collected and well-built models should be built. Here are my acknowledgements and my references. Thank you for listening.
Okay, great. That concludes all our presentations today.